Then I'm going to talk a little bit about challenges and opportunities, and what I hope to be a call to arms to all of us, whether you're a biologist or a visualization person, and then maybe one or two shameless plugs, and okay, maybe a little bit about Cytoscape in there. I can't resist. Okay, so what's network biology? This is, I did a bunch of Googling and tried to come up with the definitive term, and the answer is there isn't one, so this is mine. It's an emerging field that has bits and pieces of a number of different things in there. It is not systems biology. It is not bioinformatics. But it uses all of these different pieces. Is there value? I think when we start talking about some of these terms right away, we want to think about, is there any value here at all? This is an image from Charlie Boone's SGA data. It has already been proven to be extremely valuable in terms of, this is a genetic landscape of the cell, and the highlighted areas are showing strong interaction within certain pathways. We would expect that, but this is really nice confirmation of that. This is a recent paper that's been published that used a network approach to explore HIV-human protein interaction. And out of this, we found some really interesting new targets for the VIF protein and how it was hijacking a human protein that we did not expect, that we not previously know. And it looks like this may be a potential candidate for therapeutic. This is one that we just heard about. This is the glioblastoma set from the CBio portal. And this came out as part of the TCGA, and we can map data of all different kinds onto this to explore cancers. So another way to measure this, this is the original Barabasi paper, over 3,000 citations. This is the Cytoscape paper, a tool, and it has over 3,000 citations. So people are using this. People are recognizing its value. Why networks? They're just useful. It's a great way to express interaction or relationship between things. There's some useful math behind them that we can apply. And they're easy to augment, I will assert, and we can talk about that as we go through. And what I've found, at least in working with our collaborators, is most biologists intuitively understand them. If I put a network in front of somebody and I say, this is the protein and this line represents the relationship between proteins, and it's like, oh, okay, I get it. And I just want to emphasize, this is not the only visualization that we use in this area. It's just really a useful one for anchoring some things. Okay, so here's the taxonomy. For those of you visualization people, this is a pathway. Okay, everybody get it? What's different about this? Is this an accurate map? Answer, no. Right, it is not an accurate map. When you think of pathways, they are not accurate maps. Okay? They are necessarily a representation. And we can look at these, whether it's a signaling pathway, metabolic pathways, or regulatory pathways. All of these are really, they're a condensation of knowledge. They give you landmarks to follow. Okay? So something to think about. That's one important kind of network that we keep thinking about. The other one is interactions. I found this and I love it. Okay? This is a social network, literally a social network. And when you think about interactions, protein-protein interaction, it's very similar to this. And in fact, a lot of the techniques that we use came out of the social networking community and internet community too. There's the hairball. This is protein-ligand interactions. These are both genetic interactions. One is from the Boone lab, SGA. The other is from the Krogan lab, EMAP. They're the same kind of data, very different visualizations, and they can be mapped. And of course, the other one's similarity. I don't know if people are familiar with topic maps, but what a topic map is it's just you get a keyword in a paper and you find other similar papers. Similarity, protein-protein similarity networks, chemical similarity networks, and co-expression networks. A lot of people talk about wanting to get networks when they have expression data. And I think often what they're really talking about is co-expression networks. Very useful, and they are a similarity network. They're slightly different than a protein-protein interaction network. OK, 3 kinds of networks. There may be others, but we're going to work within those 3. So what can you do with a pathway? What I'm going to say, generally what you want to do is you're going to map data onto it. Okay? If people have spent many, many multiples of hours putting this pathway together, they tend to be hand-curated. The positions are very carefully designed. If you push the automatic layout button on that pathway, you've destroyed a fair amount of work, and you're not going to get something meaningful. You may also want to augment them. So I'm going to look at this one example. So I just love this example. This is great data. So this is the GBM pathway. Okay. What I've done is I've brought up the heat map for all of the patients on all of the genes in the GBM pathway. I've selected 2 genes. Those have highlighted in the pathway. So I've got selection back and forth between these. I can begin viewing this data set from multiple angles. And then if I zoom in on that, I can see the actual patient profile. Okay? This is easy stuff. We should be able to do this with all of our networks, right? We're just mapping data on. We know how to do this. So this is, a kind of a standard that you might want to do. You can see that in the other case, there's a single patient coloring that's showing you that. We could map that on. We can animate these things over time. All of this boils down to augmenting a network by just mapping on other data. And pathways are really useful in this context. What are the challenges? If I take a node in a pathway and search a database and find all interactors and bring it down and add it to the pathway, now I'm going to have a pathway with a mini hairball. A whole bunch of things hanging off the side of it. It'd be really great if I could do that and somehow have something which would only lay those out and lay those out in a pathway style. That would be great and something useful to see. There's a bunch of automatic pathway tools that are out there that do this stuff, but they're not integrated into the standard network visualization tools. They're kind of a different world. The big thing is information density. I have a node in the pathway. I'm sort of constricted as to what I can do with that node. I want to map lots of data onto that, short of having a bunch of ancillary windows and alternatively mapping them in and out. How do I do that? En route that we heard about yesterday looks really interesting. And I'm interested in seeing where we can go with that in a pathway context. That's probably the most interesting approach that I've seen in a while. And obviously, pathways aren't static. We know about that. That's another thing that we're going to want to see. Here's an example, IDH2. Everybody should be aware that this is involved in certain kinds of cancers. We know that. What we'd like to do is explore this a little bit, explore what happens. If I pull down one single node, I had to hand do that and get it in just right. It'd be great if this was more automatic. The problem is this tells me a little bit of information, but now what I really want to know is what changes between this pathway with IDH2 unmutated and the pathway with IDH2 mutated, right? That's a piece of data that I don't have. I don't have a good way to visualize that on this. OK, interactions. What are the things you can do with protein-protein interaction networks and other kinds of interaction networks? Obviously, layouts. Everybody should be familiar with force-directed layouts. There's many others. You can do all sorts of network analysis. There's a whole slew of topology measures that you can use to find out if this is scale-free, where the hubs are, where the node centralities or essentialities are. There's great tools for that. You can do clustering. You can do visual mapping, all of these different things. I'm going to focus on 2, clustering and data integration. So, for those of you who hate hairballs, everybody's supposed to be groaning right now. Is there meaning here? Well, a lot of people will look at that and say, oh, it's a hairball, I don't see anything. But if I take that same network, okay, and color it based upon a clustering algorithm, okay, what you can't see are the way that the lines in between, in this case, doesn't quite show up. But now, all of a sudden, I see structure to that. It's not just a mess. There's a definitive structure there. And once I know what that structure is, now I can dive in, I can carve pieces out, and I can do differential analysis of that. So I don't think we should be throwing out hairballs or standard protein-protein interaction networks right away. However, what I will say is this isn't the way I would actually normally look at this data. What I'd normally do is actually look at it in the clustered view. Okay. And so this is the clustered view of that same network. And in fact, this is a heat map of genetic interaction data. And this has got the same gene, or a strongly overlapping set of genes, between the genetic interaction data and the protein-protein interaction data. I now have two orthogonal data sets. Okay? If I then take and highlight in my genetic interaction data a cluster, okay, well, these are tightly interacting genetic. These are both yeast, by the way. You can see that highlighted. This is the pre-folded complex. And lo and behold, down there, what we see is those same genes are highlighted in the protein-protein interaction. And now I have two different ways to say, yep, these are tightly bound. If I didn't already know, if these weren't already annotated as pre-folded, I would start really looking for what kind of complex I have here. It's really good indicators. OK. So what are the challenges? Boy, we could use people to do layouts all day, all year. A lot of people would really like to see more layouts, more effective layouts. I'd love to see a layout that took the clustering that I did and explicitly factor that in to the force-directed. So take that into account in my force-directed layout. Clustering approaches. Most of our clustering approaches today that we use on networks are partitioning. So a gene or a protein, either one, is either in this cluster or this cluster. Well, we know very often that just doesn't make sense, right? A protein can participate in two complexes over time, right? So why do we use algorithms that insist that it's in one or the other? So I think we should be looking at fuzzy clustering and specifically looking at clustering over time variant approaches and some things like that. Network comparisons. One of the things that we really want to be doing is comparing networks between states. Okay, so I have a state of tumor, not tumor, right? How do these two networks change? Species between, there's some really nice work that Krogan and Eidecker did comparing Cerevisiae and Pombi. Okay, really nice. They showed how a portion of the network was rewired by this network comparison. Interaction techniques. As these things get bigger and bigger, we can't look at them all. We just can't. We need to be able to have abstraction, be able to do incremental loading, some sort of progressive disclosure. And yes, there are alternatives to node link diagrams. Biofabric is one. The other one is symmetric heat map. This is that same network. And in fact, the original paper that published that data, this was the published network, quote unquote network. And there's a lot of things to say about this. One of the things that I like about this is if what you're really interested in is complexes and clusters, it's really easy to find those. They're the blobs, okay? That tells you pretty clearly there is a cluster there in your network. So depends on what you're interested in. This is fairly useful. Okay. And it stopped. Everything stopped. There we go. OK. So similarity networks. Edge-weighted layouts. In similarity networks, also true in protein-protein interaction and other kind of interaction networks, you really want to consider the weights of the edge because you're talking about the similarity. So it's really important to take that into account. Skipping ahead, I think what I'm going to really focus on are these three. So let's take a look at the first one. This is the enolase superfamily. This is huge. I can't remember how many nodes, but I think it's like 5,000 nodes. It's a gigantic network. But what's really cool about this, if you look at it, is if I come through and I take a look at this group, and the colored areas are functionally annotated. We know what these proteins do. The gray are not. They're unknowns. This is a fantastic tool for annotation transfer. At least you get an idea. You can start doing other kinds of in-depth analysis here. And this is used by Patsy Babbitt's group at UCSF. They do a lot of work on this. OK. This is a chemical similarity network. This is exploring the availability of over-the-counter chemistry for attacking malaria. Okay? So all of the smaller nodes are available inexpensively, commercially. The larger nodes are hits for an assay for malaria. We're going to zoom in on that one. And you can see that we can begin mapping. OK, now we're mapping the 2D structures on there. We can look at those. But if you're a cheminformaticist, that's not enough. Right? This is great, but that's not enough. What you really want to be able to do in addition to that is I want to bring up all of my chemical descriptors in a table. This table needs to be linked to the nodes so I can see all the similarity. And I want to be able to calculate things like the maximum common substructure of that so I can begin figuring out and start doing lead refinement. All of that is sort of helps that I can hang this off of a network. That network gives me a lot of power to move forward with a lot of this stuff. All right. There are many challenges with similarity networks. The biggest one is the sheer size. If you're thinking of doing a layout of the protein universe, even a large superfamily, you're dealing with tens of thousands of nodes and millions of edges. We're going to have to do something like progressive disclosure. We're going to have to be looking at various ways of doing aggregation and abstraction. And at the end of the day, all of this is n-dimensional. Right? When we do these layouts, they are, in fact, an optimization. They're not real in that sense. So we've got to get other ways to make it better. And this is sort of my favorite example because I can now render this on my laptop. It takes about 30 minutes to load and render. And that's with a laptop that has 8 gigabytes. This was originally done on our cluster. So, call to arms. All right. If you're a member of the visualization community, don't stop innovating, but please stop reinventing. Right? If you've got a tool that nobody else has, you've got a new way to look at something. If you go off as part of your PhD or part of your postdoc and implement this off in a corner, okay, it may be the greatest thing, but eventually you're going to go on, and there's a fairly good chance that that's just going to go away. And we, the rest of the community, lose it. So there's a lot of existing platforms that really want your tool to be part of it. And you can publish it and everything else. There's not a problem with the publication of that. So please, there's a lot of open source tools that you can integrate with. Do it. Don't reinvent. Biological community. We need feedback. Often you sit, and you'll link together tools that you learned as a postdoc, and there's lots and lots of Python scripts and Perl scripts, and you kind of link them together, and that's the way your job is done. Doesn't have to be that way. You need to be complaining to us, the tool writers, and say, I spend this many hours doing this. Can you help? And then be willing and ready to be our beta testers. If we build it for you and go off in the corner and work on it, then we may wind up something, we'll likely wind up something that won't be as useful to you as we think it is. Okay? So again, yet another piece of effort, piece of work that goes nowhere. Okay, on to the plugs. So if you're looking for an open source package and you want to contribute some great new idea for visualization, I have one I can mention. The Cytoscape is a consortium. It's open source. There's lots of opportunities for you to help with the core if you want to do that, get right in. Or there's a great plugin architecture. And there's a rich history of publishing Cytoscape plugins that add significant capabilities. So you can write it as a plugin and get it published. We have an app store. We'll distribute it for you. We'll track downloads. It's got all of the social media kinds of things on there that people can make comments and say to the world how wonderful this is. This is my page on that app store. Yes, I have 1 or 2 plugins. But if you go through, I track all of the downloads by going to the App Store, and that's included in our annual report to NIH every year. And that's the way I get this done. So please consider this. If you have a student, particularly a really bright undergrad, who has no funding for the summer and would really love to get involved in something like this, then you should know about the Google Summer of Code. The National Resource for Network Biology has just applied to be a mentor organization again this year. This is the coolest program in the world. Google pays your student to work on our software. They can publish it. They can put it up. There's no restrictions there, but they get paid to do it. There's the URL. Check it out. We hope to get about 10 or 12 students this year. And the pay is apparently pretty good. Okay, and they get paired with somebody from the Cytoscape core team. Okay. Oops, I don't want to go there. Acknowledgements. Wow, what a list. The RBVI team, I'm a part of the RBVI, and Tom already listed all of the members earlier in his talk. I also collaborate very strongly with folks at the Gladstone Institutes. Alex Pico, who gave a talk here last year about pathways and Wikipedia in particular, WikiPathway in particular. Samad, Alan Kaczynski at Agilent Research is part of the team. The Krogan Lab are great collaborators of ours, and we do a lot of work with them. And the whole Cytoscape core team, you can see that we're spread all over the map. And also funding, thanks for keeping us funded. So with that, I'll take questions.